In 2007, the European Space Agency bolted a container of living animals to the outside of a satellite and launched it into low Earth orbit. No cabin, no pressure suit, raw vacuum, unfiltered solar radiation, the full hostility of space. 10 days later, the satellite came back, and most of those animals were still alive. They were tardigrades, and they are about 0.5 millimeter long. You have probably never seen one, but they almost certainly live in your backyard. When conditions turn lethal, a tardigrade pulls its 8 stubby legs inward, expels almost all of its water, and collapses into a tiny dried pellet called a ton. Metabolism drops to near zero. No movement, no detectable biological activity. Just a speck, waiting. On the Photon M3 mission, the vacuum of space did not kill them. The radiation damaged some populations, but not all. When researchers rehydrated the returned specimens, the animals woke up. Just like that. Scientists are now seriously asking whether organisms in ton state could survive interplanetary travel, hitching rides on meteorites between worlds. That question has a different weight when you know the answer might already be living in your garden: moss. Every winter across much of North America, the wood frog does something that should not be possible for a vertebrate animal. Its heart stops. Ice forms between its cells. Brain activity flatlines. It becomes, for all practical purposes, a frozen block. Then spring comes, and it hops away. This is not deep hibernation like a bear, which keeps its heart beating and its temperature above freezing. The wood frog, Rana sylvatica, genuinely freezes. Up to 2/3 of the water in its body becomes solid ice. Before the cold arrives, the frog's liver floods its cells with massive concentrations of glucose. That sugar acts as a biological antifreeze from inside the cell membrane, preventing the destructive ice crystals that would otherwise shred tissue. The ice that does form collects in the spaces between cells, where it causes less damage. The frog can stay like this for weeks. When temperatures rise, the ice melts, the heart restarts, and normal function returns within hours. Researcher John Costanzo at Miami University in Ohio has spent decades documenting exactly how this works. The reason it matters beyond biology is this: keeping human organs viable outside the body is one of the hardest problems in transplant medicine. And this frog has been solving a version of that problem for millions of years. If you boil pasta, the water temperature is around 212 degrees Fahrenheit. The Pompeii worm does not live near that temperature, it lives in it. Or close enough that the distinction barely matters. Discovered in 1980 along the East Pacific Rise, Alvanella pompeiana clusters in tubes built onto the walls of hydrothermal vents, deep on the ocean floor, where superheated water vents from cracks in the crust. Temperature readings inside those tubes have hit 176 degrees Fahrenheit. That is hotter than most proteins can hold their shape. Most complex animals would die within seconds. The Pompeii worm does not. Its back is covered in a dense fleece of symbiotic bacteria, and researchers think that colony plays a key role in the worm's survival, possibly insulating it, possibly providing nutrition, possibly both. The exact mechanism is still actively debated. What is not debated is the result. Alvanella pompeiana is officially the most heat-tolerant complex animal ever confirmed by science. The Pacific seafloor contains thousands of hydrothermal vents that have never been observed by humans. The Pompeii worm was waiting to be found for millions of years. Whatever else is down there has had the same amount of time to find its own arrangement with the heat. A scorpion can eat one insect and then wait an entire year before it needs to eat again. On the night it finally does hunt, it operates at full speed, with the same reflexes and the same venom delivery it would have on any other night. That is not just impressive, it is almost mechanistically strange. Research documented by Philip Brownell at Oregon State University found that scorpions can suppress their metabolism down to around 3% of normal operation. 3%. Your metabolism slows maybe 10 to 15% when you sleep. A scorpion's drops 97%, and the animal remains aware, sensing its environment, ready to respond. The moment prey is detected, metabolism ramps back up within hours. There is no groggy warm-up period. The switch just flips. This flexibility almost certainly explains why scorpions have survived essentially unchanged for roughly 430 million years. They predate the dinosaurs by a very wide margin, and outlasted them by doing the simplest possible thing: needing almost nothing. When resources collapse, most animals starve or migrate. The scorpion just turns itself nearly off and waits for conditions to improve. There is a lesson in that approach that extends well beyond biology. Picture a brine shrimp egg sitting in a dry salt flat. Completely desiccated, no oxygen, no water, baking in the sun. Now picture that egg 25 years later. Add water. The embryo inside does not start over from the beginning. It picks up development from the exact cellular stage where it stopped a quarter century earlier, and continues as if nothing happened. Brine shrimp from environments like Utah's Great Salt Lake produce cysts capable of entering cryptobiosis, a state where every biological process not just slows, but fully stops. Metabolism stops. Cell division stops. Time, in any meaningful biological sense, stops. The cysts survive complete dehydration, near-zero oxygen environments, and radiation doses that would destroy most other living cells. When water returns, the clock restarts. The embryo resumes. Researchers have documented this pause and resume biology for decades, with pauses verified at 25 years and counting. The reason this matters beyond Great Salt Lake ecology is straightforward. One of the hardest problems in long-distance space travel is keeping biological systems viable over enormous timescales. If you want to understand how that might work, the brine shrimp cyst has been running the experiment for a very long time already. In 2016, a team led by Julius Nielsen at the University of Copenhagen published a paper in the journal Science that stopped a lot of people cold. Using radiocarbon dating on the crystalline lenses of Greenland shark eyes, tissue that forms at birth and never regenerates, they calculated the age of multiple specimens. One came back at roughly 392 years old. That animal was born sometime around the early 1600s. It was already more than 270 years old when the United States declared independence, and it was still alive in 2016. Greenland sharks live in the cold, deep waters of the North Atlantic and Arctic Ocean. They grow at approximately 1 centimeter per year, which is slow even by shark standards. Nobody had previously understood what that growth rate meant over a full lifespan. They do not reach sexual maturity until around age 150. Let that settle. An animal that is not reproductively mature until it is 150 years old. A Greenland shark born today will almost certainly still be alive in the 2200s. The ocean these animals move through has changed dramatically across their lifetimes, and they carry no record of what they witnessed, which is somehow both frustrating and oddly moving. When the rivers of West Africa dry up in the hot season, the West African lungfish does not die, and it does not migrate. It digs. The lungfish burrows into the riverbed mud, coils into a tight ball, and secretes a mucus cocoon around itself that locks in just enough moisture to stay alive. Its metabolism drops to a fraction of normal. And then, it waits. Documented cases have recorded lungfish surviving in that state for over 4 years. Not weeks, years. Buried in what is essentially dried mud, breathing through a primitive lung, since it has both lungs and gills, and can use either depending on what is available. When the rains return and water seeps back into the riverbed, the cocoon dissolves. The fish rehydrates, sheds significant body mass compared to when it went in, but resumes normal swimming behavior within hours. The lungfish lineage goes back roughly 400 million years in the fossil record. Its ancestors were alive before the dinosaurs existed. The ability to wait out catastrophic environmental conditions is not a recent trick this fish developed. It is the oldest survival strategy in its entire evolutionary history, and it works just as well today as it did in the Devonian period. In 2017, researcher Federica Bertocchini at CSIC in Spain was keeping bees as a hobby. She noticed that wax moth larvae had gotten into her hive and chewed through the beeswax combs. Annoyed, she collected the caterpillars in a plastic bag. Shortly afterward, she noticed holes in the bag. The larvae were chewing through polyethylene. That observation led to a study published in Current Biology that found something surprising. The larvae were not just chewing plastic, they were chemically breaking it down. Within hours of contact, an enzyme in the caterpillar's saliva was oxidizing the plastic, beginning the same degradation process that normally takes decades in a landfill. The connection makes chemical sense. Beeswax and polyethylene plastic share structural similarities at the molecular level, and the larva's digestive system evolved to handle one of them. It turns out it can handle both. Crucially, researchers found that isolated saliva without the caterpillar still degraded the plastic. The enzyme itself is doing the work. Scientists are now working to synthesize that enzyme at scale. The goal is an industrial process that could break down plastic waste without needing millions of caterpillars to do the chewing. One distracted beekeeper, one accidental discovery, and a potential tool that might change how we handle plastic. The animals we overlook have been quietly carrying solutions we did not know we needed.